That was tricky.
Hello?
Hey,
Oh, did I interrupt something?
a little.
Oops. Sorry. You want me back, go out for a little bit?
Okay, thanks.
No, we were just kind of talking about like how we all did on the last exam. So
Ah,
yeah, and it's nice that he kind of throws a bone here or there and stuff, like spaghetti loop. I was like, what the heck is spaghetti loop? But but sometimes
Professor.
sometimes I just get to a point
Oh,
where oh, it has to be one of these two, and it's literally like just a flip of a coin. Sometimes I'm like, damn. So
yeah, I mean, that's part of it, to be honest. Sometimes you're not going to know everything, right? So you want to
yes,
make sure that you
but I
increase
think
the odds as best as you can of guessing it
yeah,
correctly.
yeah, but I think that's the nature of multiple choice, anyways. Just kind of
Mhm.
get it down to the two most possible, and then kind of see if you
Well,
get lucky.
the cool thing about, well, actually, some people like it, some people don't, about having these exams online is you can do the multiple answer questions. And so rather than it being you either get the points or not, now you can get some of the points. And so I find that's actually helped students quite a lot
that's what I
comparing.
was gonna ask too. So
Yeah.
like I know like in math, you get automatically partial credit, but like I didn't know if we get partial credit for the answers that we put that you have to select multiple of them in
Most
the next.
certainly you do, yes. And they're
Okay.
usually split evenly. So if
Okay.
there are, which is actually a bit of a pain because if there are three possible or there are three correct parts to that answer, then I have to figure out how to make them add up to 100 because Blackboard really hates like partials. No, Dipper, you can't. So typically the penalty for incorrect answers is usually equal to about one, give or take one, maybe one and a half correct answers. So let's say you've got four possible answers and three of them are correct. The incorrect one will give you a penalty of usually like 25 to 30%, something like that. So the thing is here, I don't want to be like punitive. I don't want to punish you for getting the wrong answer, but also I don't want multiple answer questions to be, well, just select all of them, right? Which is essentially the outcome is if there isn't any downside to doing so. Right. So if there's no penalty for getting something wrong in a multiple answer, every time I put one, all you have to do is just select all of them and you'll get it completely right, which isn't really getting what I want out of that question. I want to see if you actually understand what I'm asking you, and if you can just select all of them and still get it right, uh, that's not doing its job. But I don't want to screw you over, right? If you like aren't completely sure and you put one down and it's actually not the right one. Um, at most, you'll lose the equivalent of one of the correct answers. But usually it's 20, 25% off, and you can never go below zero.
Mhm. Yeah, no, that's why I was asking because like how you have that feeling to where you're like pretty confident and you're pretty sure this is the right answer. And then, but the thing is, you don't know if you got it right or wrong.
Right.
That's
So,
what really got to me.
yeah, on the multiple answer questions, usually, I mean, not always, as a kind of broad statement, say there are three possible choices. Students will almost always get one or two of them, right? Because it's kind of common sense, based on what you already know. The third one may be something that you'd have to kind of think really carefully about and really understand, like what I'm talking about, like in terms of the concept or the process. And so for that third one, if you're really not sure, then I mean there'll always be two potentially correct answers, right? And a multiple answer. Otherwise, I'll just do a multiple choice. Um, if you're really like 100% on two, and then of the remaining two or three, one of them you're like, maybe, maybe not, then it's a judgment call as to whether you take that chance and you click it, or leave it and be
But
happy
that
with
could
what
be
you
a
get.
life or death situation for some grades.
Hopefully not life and death. I
It's
don't want.
horrible. It's
I don't want.
painful.
Yeah, I know. It's a tough course, and it stresses students out a lot. But also, that's the purpose of the lab and the homeworks, is to kind of take away a little bit of that stress. You know, that's kind of why they're there. So it's not all about the exams. So you can actually get a consistent, like, solid D grade in your exams. And if you do everything you should in your homeworks and your lab, you can still pass the course with a C.
Now, I had a question. So I don't
Not that I
know
want to
if,
suggest doing that, because obviously that's not what I'm after,
but pertaining to the labs,
Mhm.
are those, or in those classes that you teach, do you usually drop the lowest lab grade, considering that we have
Nope.
so many? Oh, okay.
There's always actually, I've got to, when the email comes around, I'll send it out. The extra credit opportunity here is filling out your student, well, what's the R stand for? I can't remember. SRIs. So I always give what would it be? 10-point extra credit. It's like about half of an exam grade, basically, if 80% or more of the class fill out their SRIs. So that's like a freebie. That'll bump you a little bit on one of your exams, basically.
Okay. So it's just applied to the class portion, the SRI.
Yeah.
Okay.
Oh, actually, no, it's applied to the both, because they are yeah, they're separate. So the lab and the I think the lab and the lecture are separate SRIs.
That makes me feel a little bit more comfortable.
Yeah, it's just a little bit of a boost. It's not going to make the difference between you getting an F and a C. But if you're a little bit on the margins, then let me just type this out again. Would that be lecture and lab? So thank you for reminding. I was actually going to email that round when I get the email saying that they're available, but I'll do that as well, send an announcement. And also there's a small rounding deal as well. So usually if it's, let's say you get 69.5% overall course grade, then that gets bumped up to a C.
And if you don't mind me asking, what is the test average score?
Oh, sure. I haven't actually calculated that.
Now I'm really nervous.
Might be great. I don't know. I've been so rushed off my feet this summer. I haven't actually spent a whole bunch of time analyzing the exam data. Typically from memory, it's usually like a low C, kind of between 70 and 75% typically, but it's got a really wide dispersal. And so that also includes, students that don't take it. So
and you don't do like a curve or anything, or I don't know if that would if that would benefit us or not.
No, I don't tend to curve. I'm very deliberate about doing that in the exam itself. So in the exam itself, there will always be a whole bunch of questions which are just simple recall, lookup type questions. And so that's essentially that's the curving, right? And then the trickier questions, not trickier, shouldn't use that word, harder questions make up a smaller portion of the exam. And there's even like the occasional true false question, right? You got 50% chance of getting that right. So yeah, the kind of the curving, I guess, is done in the design of the exam. So even though the questions have equal weighting, like in terms of points, they don't have equal difficulty. So the hard ones aren't going to screw you over, in a sense. Well, not
Right,
too
and then
much.
for those SRIs, right? If we all kind of give you 5 stars, right?
No, it's absolutely not about what you put in them. So the reason why we do this is actually something that my colleague instituted when he taught genetics, is that it's really good to get feedback from students to figure out things that work and things that don't. And also these are used in our evaluations as faculty, also whether or not we get tenure. So kind of my job is on the line, which is important that I do a good job. And so we really want honest feedback, you know, and typically if you don't kind of give students a little bit of a, you know, like free pizza for doing so, you'll either get people who absolutely love me or who absolutely loathe me. And those kind of extremes, it's like reading Amazon reviews, you know, you kind of also want some of the ones in between too. And hang on a second, Adam. So that's why the 80%. It's not a like a bribe. It's a to encourage a useful sample size, essentially, is the purpose for it. Because otherwise you tend to get around 50–60% of students. Oh, I have never looked at my Rate My Professor, and I never will. Never, ever, ever, ever. In fact, actually, I was kind of getting a little bit off into the weeds now. I was at a worm conference. So I work on worms, C. elegans, and it's called informally the Worm Conference. And there's actually, because we're goofy scientists, right? There's actually and I'll get to your question in a moment, Adam. What's called the Worm Art Show or a Worm Comedy Show. There's actually an art show, which I've won. And a comedy show. And in one of the worm comedy shows, I remember they actually said, oh, and now for the leading professors in the C. elegans field, we're going to look at their ratings on Rate My Professor. And so they actually brought up like Rate My Professor on this big screen at UCLA, like kind of amphitheater, whatever it's called, and started typing in like one of the top guys in the field. You could hear this collective intake of breath for like all the professors that are in the audience. It was pretty funny. So yeah, never looked at those, never want to. Hopefully they say good things, but maybe they don't. I don't know. So Adam, test 3, terminology, principles, concepts. So that's the deal with 3 and 4, is that there will be a lot more detail stuff because as they say, the devil's in the details. So to really understand like translation, you have to understand what I put on the board, right? You have to understand the role of translation factors in bringing in TRX, pardon me, GTP in translocation, stuff like that. So there's less math. In fact, almost no math. Yay. But there is a whole bunch more detail. And so it's the details that tend to trip students up. They can get the general idea of what's going on, but if you kind of delve in deeper, like, well, exactly how does the release factor work? Then you need to know that, because otherwise it's just kind of words, right? Describing stuff. Oh, don't do that, Robert. I don't want to know. I really don't. There you go. I'll give you an insta fail if you tell me. Ignorance is bliss, as far as I'm concerned. Anyway, so that aside, rest in peace, Robert. Anybody have any questions about the stuff we've covered for exam 3? Like things that still don't click, make sense? stuff you want clarifying, things like that. Because now's a good time to ask.
So in translation, we talked about the eukaryotes and prokaryotes, but we don't need to know anything about like viruses and stuff, right? Because they're kind of...
Well, viruses use whatever the host machinery is, right? They don't typically encode very much of the machinery that replicates them. They hijack the host machinery to do that for them. So yeah, essentially it's the same, right? The only real differences with viruses is when you have like funky stuff like retroviruses, like HIV, they encode a reverse transcriptase to turn RNA into DNA, for example, like retrotransposons do. And the reason they encode that is because we don't have that. Most eukaryotes don't do that. So they won't have that gene or that protein for the virus to use.
So just to make sure I know this correctly. So the central dogma in genetics, first, it starts off with the replication process, and that's where the DNA is split into two strands.
Nope. Transcription. DNA replication is a whole different kettle of fish.
Okay. So it's transcription, and then the processing from that to translation in the ribosomes.
Mm-hmm.
And then from there, how will we know whenever it's fully done and made a protein again?
When it's functional.
When it's functional. Okay.
Yeah. So we haven't covered post-translational modifications yet. That's going to be a little way down the line when we talk about control of gene expression in eukaryotes. So don't worry about that for now. We're just really covering the mechanisms of transcription, RNA processing in eukaryotes, and translation.
And then transcription is first started off with the promoters, correct?
That's right.
Okay. Yeah, sorry, just because like I took your notes and I also referred back to some old videos that I learned from in Gen Bio 1, and I'm just making sure I got them down.
Right. It's very common for students to confuse DNA replication with gene expression because some of the principles are the same, splitting the DNA apart so that you can get access to single-stranded DNA. And quite often students confuse transcription and translation as well because they sound similar and they're all involved in producing a protein. It's just there are different steps in that path. So always remember that translating is turning it from one thing into another. So we're turning it from nucleic acids into protein. Transcription is copying the information in DNA and making it into RNA.
And then whenever in the transcription phase, the messenger RNA, mRNA, has two different ends, the 5' and then the poly A tail.
In what type of organism? Here you go, here's a question for you.
Eukaryotes.
Exactly, good.
But in prokaryotes, is it just the 5'?
They don't cap or tail their mRNAs. I don't actually know why they can get away with that, whereas eukaryotes can't. But they don't. Here's another question. What else is the 5' cap used for other than, and actually the poly A tail too, other than just protecting the ends of the mRNA in eukaryotes? What else are those two structures important for?
Isn't it for like the binding of the new, like the caps will cut, if I'm thinking this correctly. No, that's splicing, right?
Yep. Splicing is also part of RNA processing, but it doesn't have anything to do with either stability or what I'm talking about. So what's the 5' cap? And also the poly A tail, really important for, in addition to stability. Think through the process. What comes after mRNA processing in eukaryotes?
Isn't it the folding?
Nope.
No.
Well, I mean, technically, export from the nucleus to the cytoplasm comes after processing.
Oh,
What comes after that when it's in the cytoplasm?
it's a good
What's
question.
the next step? It's central dogma. You just told me. Transcription, RNA processing, at least in eukaryotes. What's that one? What's the third step? Also begins with a T.
Is it the transfer
Mm,
mRNA or
that's involved in it. What's the whole process called?
translation?
Right. So what role do the 5' cap and the 3' poly A tail play in translation? So this is kind of what I'm talking about in terms of the exam. This is the kind of the details that are important to know.
You
Let's
got me.
just start with the 5' cap. So in eukaryotes, how does the small subunit find the start codon? What's the process? What are the steps?
Well, first the mRNA will be processed through the APE.
Oh, no.
No.
Talking about initiation, we're not talking about elongation.
Oh,
How do you initiate translation in eukaryotes? So obviously it has to start at the start codon, the AUG. That's where translation starts, but how do you initiate the whole process?
gosh, I'm trying to think.
Okay. These are the details that you will be quizzed on. In fact, from memory, at least there's a couple of questions, if not more, on this.
Does anyone else know? I'm still trying to think.
Okay, so true or false? In eukaryotes, the small subunit assembles on the start codon. In eukaryotes.
False.
Okay, so where does the small subunit
Because
first?
it went like the first cap, the leading part to where,
You're getting there.
yeah.
I think all the information's in there. It just needs a little bit of sorting out and kind of clarifying, right? So the 5' cap recognized by 5' cap binding proteins, right, which are initiation factors. Those then recruit the small subunit to the 5' UTR, which I think is what you're thinking about in terms of leader RNA. That's actually a different kind of term, which we shouldn't get confused with. But the 5' UTR, that's where the small subunit assembles, right? And then it uses the FMET tRNA as a little barcode scanner, right? So the small subunit then leaves the 5' end and goes dun, 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 along until the tRNA that's in the P site finds the start codon. Then what happens? We have the small subunit, we have the tRNA, mRNA. What else do we need? If we have a small subunit, most certainly we're going to have a what size subunit?
A large subunit, and that's where the polypeptide chain forms.
Correct. So the large subunit is the one with the A, P, and E sites. So that's what does the peptide bond, magic. The small subunit is all about the mRNA. That's what moves along the mRNA. Okay.
And then, sorry.
Oh, and also before I forget, and also the mRNA bends round, and the 3' poly A tail, or at least proteins that bind to it, also help with initiation of translation. It's not quite as clear how, but it also plays a role in that. So that's going back to the original question. What other roles do that 5' cap and the 3' poly A tail, in addition to stability, well, they're involved in translation initiation as well.
Okay. And then my other question was going into the splicing. So SNRPs, we had said that they're nuclear ribonucleoproteins, correct?
Particles at the end of that as well. Yeah. Just keep adding words and you'll probably get it right.
Okay. Let's see.
Small nuclear ribonucleoprotein particles. Yeah, that's the full word. Yeah. Anyway, SNRPs.
And then a combination of the SNRPs with the other proteins is going to be the lysosomes, correct?
Well, yes. So the SNRPs are complexes of RNA and protein. So there's small nuclear RNAs, snRNAs, and proteins. That's what makes them ribonucleoproteins. Those are the things that do the cutting, bending, and sticking in splicing.
Okay, so you said small nuclear RNA with nuclear proteins. And then the lysosomes are the ones that actually do the editing.
Correct. Those are the cut, bend, and stick complexes. 5 of them from memory, at least for the normal type of splicing. So funny. Every time I talk about splicing, I always think of the scene in Fight Club. When I don't know if anyone else has seen that.
Hey, no one else talks about Fight Club if it's not Fight Club.
Right. Yeah, anyway, always just makes me laugh. Like a little titter going on in the back of my head.
And then the product of, is it the proteins that are spliced together are known as exons, which are considered
mRNA.
Oh.
Protein coding, but they're still mRNA at that point. Well, so it stays mRNA, doesn't change.
The product of splicing together mRNA are exons.
Well, the exons are always there. What you're doing is you're removing the bits in between them, which are the introns.
Okay.
So you have one continuous protein coding sequence rather than chunks. You've got to take out those bits in between because otherwise it's not going to work right.
And then introns are just pieces that are really trash and don't matter, and yet they interfere with the segments.
Well, that's a little bit derogatory calling them trash, but they don't code for protein. I don't think we understand them well enough to know what they do, but they don't code for protein. So yeah, if you don't remove them, then ribosome isn't going to know whether there should be an intron there or not. Or it's not going to know that the intron shouldn't be there. It will just continue translating into the intron. So you'll either get a gibberish protein or you'll get a stop code on an truncated protein. Okay. Either which is pretty terrible.
Once that's done, the mRNA moves out of the nucleus into the cytoplasm where translation occurs.
That's right.
Okay. Thank you.
Cool. Good questions.
Hopefully I get a good grade on this test.
I hope so too. Anyone else have questions? I'm just having a quick look at the schedule, see what we got coming up. Holy guacamole. Just next week and the day after that. That's crazy.
Okay, I have like a question. So in translation, I mean, there's a lot of different types of RNA, but there's kind of three main ones, which is the messenger RNA or the mRNA, tRNA, which I know those two. But then there's rRNA, which is what's a ribosomal, or what was that? Okay.
Correct.
What role does rRNA play in translation again?
So I haven't posted that picture yet, but remember that kind of convoluted diagram of how elongation works?
Okay, yeah. Okay.
At the top right, I wrote peptidyl transferase.
Okay.
So the thing that cuts off the amino acid from the tRNA in the P site and then sticks it onto the amino acid in the A site, that's what our ribosomal RNA does, or at least one of them.
Okay.
So that's one of the enzymes in the ribosome. There are other ribosomal RNAs. It's not just one. I can tell you how many actually. I know there's at least two. But the main one does that amino acid transferring activity.
Okay.
And don't forget, it's a ribozyme. It's an RNA enzyme. Hint, hint, wink, wink. Also another, so this is kind of like a funny, it's funny to me maybe, but may not be to you, but whenever I have these catch up kind of, oh, cool. Thank you, Adam. That's a help. Typically I'd go over processes and facts and stuff like that in class that relate to the exam. So have a watch through of this before taking the exam. That would be my advice. And so that's also another way in which actually attending these things benefits you. Not only do you learn about stuff, but also get some focused revision, I guess. It's like an extra benefit. All right. Anyone have any other questions? Cool beans. I'll have a look at that link later, Adam. There are lots of good videos, and most of them are somewhat accurate too, online, which are worth looking at if you don't quite visualize what's going on. Cool. All right. So essentially what we've done, up till now in the course, right, is we really looked at like the inheritance of traits, like the basic patterns of inheritance, how kind of alleles get passed down from one generation to another. We've looked at the basic structure of DNA and RNA and, protein too, and how you go from one to the other. So that's all well and good, but really it doesn't capture like the whole picture. And the piece that does is how do you control all of that, right? And so really the control of gene expression is essentially the idea or the concept or even just like the biology that ties all of that together, right? And it's actually really interesting because in terms of evolution, there's a giant bun fight between a group that believes that evolution is really driven by changes in protein coding sequence, right? Which are kind of more, what would it be? Can't remember her first name. Hoekstra, I think her name is. I think she's at Harvard. And another guy whose name escapes me for now, who really is saying, well, it's actually the control of gene expression which drives evolution, right? And they're probably both right, they're both very smart people. So it's not just what protein coding sequence you have, it's also how you use it, right? And that's what this next section is largely, not entirely, but largely going to be about. How do we control when, where, and how much a gene is expressed? And so you can think about this in lots of different ways in terms of biology. Think about it in terms of bacteria, which in some ways are a simpler way of doing things. You're a bacterium. You know, probably actually using their flagella to like scoot around and, you know, wherever they're at. There are hordes, teeming hordes of other bacteria around. They're essentially looking to eat your lunch, right? If you think about it in that terms, right? Even on your skin, like your skin is covered in bacteria, good bacteria, one hopes. If they're not good bacteria, then you get things like eczema, right? Which is a kind of a bacterial imbalance in your skin, which is kind of weird. So those bacteria are basically in an arms race with each other. And so it talks about the cost of gene expression. Making, yeah, exactly, dermatitis. I actually suffered really badly from childhood eczema. And that was before they understood what caused it. Most of the treatment that I had was basically kind of treating the symptoms, not actually treating the cause.
Just a curious question.
Go on, Adam.
So like going with the dermatitis and eczema, so like I have a friend who has eczema and he's had it for a while.
Mm-hmm.
Of course it gets severe sometimes, but how exactly does it originate?
That's a difficult question. So typically asthma, eczema, and allergies tend to run together. So I have allergies to dogs and cats, funnily enough, and a little bit to all the crazy crap that floats around outside in San Antonio. But also I had eczema. And eczema is actually really poorly understood. So what they do know is that it's essentially somehow, and I don't actually understand that, there's an interaction between you and your bacteria. Right, that's the case in your gut, where those bacteria produce really important products that we can't produce ourselves, which is just beginning to be understood. So your gut microbiome is important, but also your skin microbiome is also really important. And there's crosstalk, right? So, you know, we at a cellular level tell them stuff, and those bacteria tell us stuff, and hopefully that kind of song is in harmony. But in the case of eczema, what happens is your skin gets over-colonized by different forms of bacteria, Staphylococcus, I think. I can't remember which species it is. And those are what produce the skin irritation and, you know, other unpleasantness that goes with eczema. Why the native bacteria that you're supposed to have kind of die off or are less successful, and how these other ones can kind of take over that niche, I don't actually know. I'm not sure it is known. But one of the treatments now for eczema is actually a mild bleach bath. Right, so you kind of dunk your kitty in a, obviously it's not like what you'd use to clean your bath out or drains or whatever. But the idea is essentially you kill as many of the bacteria on your skin as possible, and then you can get creams which have the right kind of mix of, you know, friendly bacteria to recolonize the skin with, right? Which is something that's really come about in the last 20 years or so, or maybe even less. Yeah, and then that's treating the symptoms, but the actual cause is a bacterial imbalance, right? And so why that occurs, I don't actually know, but there's kind of communication between us and the bacteria that make up, I think about 5% of your body weight is bacterial, right? And including guts and gut bacteria imbalances, thought to have a big role in things like autism. You know, you actually get a lot of your bacteria from your mother as you like make your exit, so to speak, and through various kind of interactions afterwards, like breastfeeding and handling, stuff like that. So, but the key point is though, is that if the, you know, the good bacteria don't do as well, other bacteria will colonize that space, and those might not be quite so friendly. And so essentially, it's like this kind of arms race between bacteria. And what you really want is for that to be kind of in sort of equilibrium, right? It's not going to be static. It's going to be constantly changing, but it's going to be in equilibrium. And so those individual bacteria, they have to be able to, A, not spend any more energy than they absolutely have to get as much food as they absolutely can, right? So really like hunter-gatherers essentially in a microcosm. So they have to be very, very, very careful not to make any mRNA or protein that they don't need. But when they do need it, go like all in. You know, I'm not going to like put in like a few dollar bills here. I'm going to like bet the farm on this. Because otherwise, other bacteria around them will be like, oh, this looks tasty. I'll have some of this. And then they'll be able to divide, and then they'll colonize that kind of niche, right? That ecological space. And that's the same whether it's on your skin, outside in the soil, in a rotting apple. Doesn't really matter where. It's the same kind of idea, right? So this is an inherently costly process. So you only want to spend that, you know, that money, energy terms, if you get a return. But if you don't spend it, someone else is going to use it, or some other bacteria, right? They don't have personalities. Some other bacteria is going to use that and divide and colonize that space. So that's from like a kind of a single cell perspective, right? From our perspective, we're much more complex, right? So we're an assembly essentially, right? If you think of it this way. I mean, you can kind of chop us up and put us through a strainer more or less, if you chop us up finely enough. But we're an assembly of millions of different cells that all have different roles. They're in different places. They respond to different stimuli, right? And so we get that way through a very, very carefully controlled process, which is called development, right? So we start as one single cell, zygote, a fertilized zygote, which divides and then divides and then divides and divides and divides thousands of times until we end up a functional human being like me. We don't end up a giant blob of cells that's just kind of like schlepping around, you know, absorbing nutrients and cats and stuff, right? So those cells have to talk to each other, right? They have to control when they express certain genes, because that is what makes them, it's what makes skin cells. It's what's make, you know, liver cells. Every single cell in your body, other than gametes, so sex cells, and red blood cells, because they don't have any DNA, or at least any genomic DNA, all have the exact same set of instructions. Every single one. So how is it that, you know, I have fingers here, I'm not going to show you my feet, I have toes down there? Why are those different? Why do I have toes down there and not toes on my hands? Right? Why is my, I like my toes. I think they're cute, short and stubby. But anyway, why do I have 4 digits and a thumb? Why is my thumb here? Why is my thumb not sticking out the other side, right? All of that is controlled by differences in gene expression at different times and in different amounts, right? You know, in response to different signals. And so really, that's what makes us who we are, right? All the information is there in every cell almost, right? But we end up these complex, fascinating organisms through a very careful choreographed almost, right? If you think like synchronized swimming or something, interplay between different cells and which genes they express and which ones they don't, right? And at which times and in how much in response to what, right? So really you can't think about anything in biology, be it in terms of ecology, in terms of evolution, in terms of development, anatomy and physiology. You know, why do blood cells form where they do and connect together? Why don't they just go off into the weeds on their own, right? You can't think of any of that sort of stuff without understanding the basics of how gene expression is controlled, right? And really, it's one of the kind of underlying, you know, principles of biology, of life itself. And I'm sure Watson, Crick, and Franklin and Maurice, yeah, I think I've got them all. When they figured out the structure of DNA and, you know, posited the sequence hypothesis and central dogma, I don't think they ever realized exactly what it was that they were discovering, at least at that point. You know, the kind of how all of that ties together to make us, you know, what we are. So it's a really, really cool subject. I love talking about it because it really underpins everything. You know, it's super, super fun. It's also bloody complicated, right? And as you would expect, it's not going to be simple because we're not simple, right? Bacterial responses to external stimuli are not simple, right? And the cost benefit calculation is also not simple. And so really like the two kind of overriding principles are bacteria control their gene expression in groups. So they typically have all of the genes that are involved in one process grouped together into operons. And we'll be looking at two examples, the lac operon and the trip operon, which is short for lactose and tryptophan, right? So the genes that are involved in the processing of lactose into something bacteria can use, and the synthesis of tryptophan from its precursors, right? And those illustrate some really kind of underlying principles, essentially, of what's going on. Eukaryotes, on the other hand, have their genes scattered all around through the genome, and they coordinate gene expression through use of transcription factors, right? So these are separate proteins which can turn on multiple genes at the same time. They're involved in the same process. So those are two very, very different ways of controlling gene expression. And also in eukaryotes, you have a whole extra load of bells and whistles and wrinkles that we'll talk about too. And it is a phenomenally complex subject, right? It's actually, without a doubt, one of the hardest subjects in this course. But it's also one of the most interesting from a kind of a broader biological point of view too. And one which you'll need when you take other courses, subsequent cell biology, developmental biology, if it's ever offered, stuff like that. What else? Also, we'll need to cover the general ideas of positive versus negative regulation, right? So turning something off versus turning something on. Inducible versus repressible. So being able to induce expression or being able to stop it, right? If it's naturally going to occur. Kind of those sort of concepts, which we'll cover really in the beginning of class on Tuesday. Kind of had hoped to get to that today, but it was good to spend more time on the questions for the exam. Make sure everybody's good on that. Yeah, and that's pretty much it. Well, I mean, that's not it. We've got a whole ton of stuff to do next week, but that's pretty much it for today. I don't really have anything else to chunt on about, and my wife has distracted me because she's cooking something really, really tasty, and I'm hungry.
So I know it's 2 weeks from now, which is crazy. But is the final exam, like the time for it, is it going to be the same? And will you have it open like one day and then it'll close the other day? Because I know, like me, I'm busy at work and I try to put time into it.
For sure.
So, still teaching.
Daughter's upstairs shouting downstairs. So time is the same. The format will be the same, both online and open one day and closes the next, to give you a bit more flexibility. And the amount of material is roughly the same as well.
But
So-
encompassing everything we've gone over.
No, it's not cumulative. It's just over gene expression, and if we get time, some fun stuff on biotechnology and that kind of jazz. CRISPR-Cas and
Oh,
cloning
okay. So
humans.
we don't
and
really
stuff.
have a final cumulative exam. Woo!
No.
Thank
No.
goodness.
Yeah, that's what
Okay,
Robert said.
don't take that back. Don't take that back.
No, and I mean, essentially, in a way, it's cumulative in the sense that you can only really understand what we're talking about with all that you learn prior to that point, right? But it's not cumulative in terms of information. Oh, I hope not, Dipper. You know, study hard and ask questions, and hopefully you'll be okay. Yeah, these exams aren't designed to punish you, that's for sure. But they
Yeah,
will really stretch you.
The reason why I was asking, because I know in like Gen Bio 1 and 2, they're all cumulative. You have your 3 exams plus a final. So that's
Yeah.
why.
Not the case in this one. So it's not technically a final. It just happens to be the last exam for the course. So it's exam 4.
Gotcha.
Cool. All right. So going to take a half-hour break for lunch and leg stretching and the like, and then we'll hit the lab at 2 as per usual. If you have any questions about the lab assignments, that'll be a good time to ask. And if you still have questions about exam 3, then ask away as well. We'll have time to do that. I don't think what we have to do today is too onerous. It'll probably be about an hour, if that. Cool. Thanks for all the questions.
I won't be able to make lab today, but will you record and post it?
Yeah.
Okay. Thank you.
I'll really get on and make sure all this stuff is posted as soon as lab is finished. Usually I have a little bit of staggering around in the days afterwards, but I'll make sure it's all up quickly for anyone that wants to take the exam this afternoon. Cool.
Thanks.
All right, everybody. I shall see you in half hour. Enjoy your lunch. See you in a little bit.